tissue distribution of rat glucagon receptor and glucagon-like peptide 1 receptor gene expression. This work will be presented by Solomon, Zinga, and Fadhiya. The aim of this presentation is to review the original research paper that stated the wide tissue distribution of rat glucagon and glucagon-like peptide 1 receptor gene expression. Recent studies have revealed important advances in our comprehension of glucagon action, especially the cloning of glucagon receptor cDNA, which is quite similar to GLP-1 receptor. Mammalian proglucagon is post-translationally processed in pancreas and intestine. Receptors are cloned only for 2 of proglucagon-derived peptides. The role of glucagon and GLP-1 in glucose metabolism is well established. However, there are other roles which need further studies, such as GLP-1 involved in the clearance of macromolecule from lung airways. This additional role indicates the wide distribution of tissue expression of the glucagon and GLP-1 receptors. GLP-2 has trophic activity in mouse intestine, and it is more likely to be the proglucagon-derived peptide which is involved in the intestinal adaptation. This study addressed the possibilities that novel but related receptors exist for proglucagon and derived peptides. Multiple isoforms of the cloned glucagon and GLP-1 receptors are also expressed. And finally, the 2 known receptors may be expressed more widely than initially suggested.
The method that is used for isolation of total RNA is guanidine isothiocyanate, cesium chloride gradient method. In the reverse transcriptase polymerase reaction, the reverse primers as well as the PCR primers were designed from homologous region between the glucagon and GLP-1 receptors. The RT-PCR products were subcloned into pGEM4Z and then sequenced by the dideoxide chain termination method using sequenase. In the southern blot analysis, the phosphorus labeled cDNA probes were synthesized from clones of the glucagon and GLP-1 receptors, corresponding to the region between the 3rd and 6th transmembrane spanning domains. Positive glucagon and GLP-1 receptor hybridization controls were included in the analysis. As the southern blot analysis was semi-quantitative, they used the nucleus protection assay to quantitate the result. The glucagon receptor cRNA probe is identical to the southern blot cDNA probe. A second GLP-1 receptor probe was synthesized from a clone isolated by RT-PCR because the GLP-1 receptor probe that was used in southern blot analysis was unsatisfactory for the assays. A cRNA probe for the ubiquitously expressed beta-2 microglobulin gene is used as an internal control.
Now let's see the results that's obtained from this experiment. Using a combination of RT-PCR primers designed from the conserved regions between the glucagon and GLP-1 receptors, glucagon receptor clones were isolated from corpus, terminal ileum, hypothalamus, and liver, whereas GLP-1 receptor clones were isolated from corpus and terminal ileum. This figure shows the results of the southern blot analysis of the RT-PCR products. The RT-PCR products were visualized on agarose gels by ethidium bromide staining, and it's presented by figure A here. Then they were transferred to membranes for southern blot analysis and presented by figure B and C. As we can see from this figure, there was a high level of gene expression for glucagon receptor in corpus, liver, and kidney, whereas a high level of expression was seen in corpus and lung for GLP-1 receptor. As we can see from this table, the southern blot analysis indicates the wide distribution of these 2 receptors in rat tissues. This table also shows the relative strengths of hybridization of the glucagon and GLP-1 receptors. there was a high level of expression of the glucagon receptor in liver and kidney, as it was anticipated in the previous studies, but also there was a high level of expression in corpus, spleen, and thymus. The GLP-1 receptor was highly expressed in corpus and lung. As the Southern blot analysis was semi-quantitative, they used a nucleus protection assay to quantize the relative level of gene expression, and they presented their results with this figure. This specific figure shows the glucagon receptor messenger RNA levels. The highest level was seen in liver, and the levels of the rest of the tissues was expressed in percentage of liver expression. And as we can see from this graph, the highest level was, relatively high level was seen in kidney tissue, which is almost 41% of the liver expression. This figure also represents the result from the protection assay, which is for the GLP-1 receptor messenger RNA level. We can see a high level of expression in lung and corpus for GLP-1 receptor. This was parallel with the results found from the Southern blot analysis.
Discussion. The very sensitive RT-PCR allowed the detection of glucagon and GLP-1 expression in a very wide range of rat tissue. The nucleus protection assay allowed the quantitation of expression and therefore more accurately reflect the level of expression. But the significance of our RT-PCR is limited because the amplification of transcripts was not shown linear. We have to say also that there is no discrepancy between the intensity of ethidium bromide stain bands and hybridization of the two receptors probes, meaning that there are no novel receptor sequences amplified. The relative expression of glucagon receptor in different tissue was similar to the finding by Hans and al. In 1995. At the end, we're going to say that the presence of glucagon receptor gene expression in the spleen and thymus is consistent with the suggestion that glucagon may play a role in immunomodulatory.
This paper clearly demonstrates the wide tissue distribution of the glucagon and GLP-1 receptors expression. The glucagon, GLP-1, and other pro-glucagon-derived peptides may have more novel functions. Last but not least, novel receptors for pro-glucagon-derived peptides may also exist. Thank you for taking your time to watch our presentation.